Good afternoon, folks. Hope everybody is doing well today. This is part 2 in my lecture series on gene expression and RNA. If you have not already watched part 1, I strongly recommend you go and do that right now so that you have the necessary background for what's going to be talked about in this section. And if you are in my biology class, I recommend you go on Blackboard and get a copy of the handout summarizing the vocabulary and the key points in the notes here, as well as including a couple of helpful diagrams that'll help you to visualize the things I'm going to be talking about. So, in our last lesson, we detailed what gene expression is, this process by which a cell decodes the information stored in DNA and uses it to make the functional proteins that it requires to carry out its functions. We laid out the central dogma describing the flow of genetic information from DNA to RNA to protein. We talked about what RNA is, how it differs chemically from DNA, and the 3 different kinds of RNA. And we laid out the process of transcription, by which data is copied from a DNA molecule, copying out the gene that we want to express to a molecule of mRNA. So that is where we left off. So we've done the process of transcription, which is the first half of our process of gene expression. So that was finished in the nucleus, nucleolus of the cell specifically, if you're a eukaryote. In a prokaryote, it's just in the cytosol like everything else. But we've built our mRNA molecule. We're going to put it through a process known as splicing, which I'll talk about after we discuss translation, because you'll need to know some things about translation first. And now we're going to take that mRNA molecule, the transcript of the gene, if you will, and we are going to move it to a ribosome. And it's at the ribosomes that we are going to carry out the process of translation. Translation is the second part of our process of gene expression, the part where we go from RNA to protein, taking the data that is encoded in that RNA molecule, the transcript of the necessary gene, and reading it so that we can build whatever polypeptide it codes for. Keep in mind, remember, I might be using the terms polypeptide and protein sort of synonymously here. Gene expression means building proteins, but remember that any given gene is just coding for a single polypeptide, and you might need more than one polypeptide to build your complete protein. So, our process of translation, like transcription, is going to be a 3-stage process. It's going to have 3 distinct phases, which we identify in the same way that we identify the phases of DNA replication: initiation, elongation, termination. That said, the events that are going to happen during translation are very different than those that are happening during DNA replication or even transcription, just because we're not actually building a nucleic acid at this point. Rather, we are reading a nucleic acid to learn about how to build the necessary protein product. So, our process begins with initiation. Initiation is the prep stage. During initiation, the mRNA molecule arrives at the ribosome. Now, the ribosome is going to have 2 components, often called the greater and lesser components. The greater one is the bigger one that sits above the RNA. The lesser one is the smaller one that sits below it. And what's going to happen during the process of translation, during initiation, the RNA is going to be pinched between the 2. We're going to kind of build the greater and lesser components onto either side of the RNA. It's like we're making an RNA hamburger with the ribosome as the top and bottom bun. Specifically, the mRNA is going to bind to the rRNA present in the ribosome. That's the entire job of rRNA, to bind the mRNA and hold it in place. We know where to begin because of a start codon. Remember how I said that every mRNA molecule begins with the start codon, AUG. So that's going to tell our ribosome, here's where you start reading this gene from. So we're going to bind to the mRNA at the location of the start codon. rRNA binds the mRNA. Once the ribosome has attached to the mRNA, we're going to begin preparing for the rest of translation by assembling all the different molecules we are going to need for that process, which are collectively referred to as the initiation complex. Things that are included in the initiation complex. First off, we're going to have amino acids. The whole point of what we're doing here with translation, after all, is we're building a polypeptide, and you know that a polypeptide is a chain of amino acids. So we're going to have a whole cloud of amino acids waiting in order to be assembled. All 20 amino acids are probably going to be present in that cloud, multiple copies of them too, because any given protein is probably going to contain several copies of the same amino acid. Then we're also going to have a series of tRNA molecules. Remember, tRNA is transfer RNA, and the entire job of tRNA, you might recall, is to move amino acids to the ribosome and put them in position. So we're going to need tRNAs to carry this process out. We're going to have a wide variety of tRNA molecules to bind the different amino acids. We are also going to have a series of activating enzymes. The activating enzymes are going to bind the amino acids to the growing polypeptide chain. There's going to be one activating enzyme for each possible amino acid. Ergo, we're going to have a total of 20 activating enzymes. And then finally, our initiation complex is going to include a wide variety of accessory proteins, helper molecules that assist with various aspects of the translation process. We're not going to go into full detail about all the things that those are doing. So that is initiation, prep stage. So in summary, mRNA has arrived at the ribosome. Greater and lesser components of the ribosome are going to sandwich around it. rRNA in the ribosome binds the mRNA at the location of the start codon, AUG. And then we're going to assemble our initiation complex of all the stuff that will be necessary to carry out the rest of translation. So that's initiation. Next up, elongation. Just as with DNA replication and with RNA transcription, elongation is the building phase. This is the phase during which we are going to assemble the polypeptide that mRNA molecule contains the code for. Here's how it's going to work. The ribosome is going to move down the length of the mRNA molecule, starting from the start codon. We're always going to move in the 5' to 3' direction. So starting at the 5' end where AUG is, working our way to the 3' end where our stop codon is going to be located. Now the ribosome is going to make its way down the mRNA. It's going to move one codon at a time. And recall that a codon is a group of nucleotides, 3 letters in our genetic alphabet, which represent one specific amino acid. So ribosome moves down over a codon. When that happens, we say that it is quote-unquote reading the codon. When the ribosome reads the codon, that tells it which amino acid needs to go in that position in the polypeptide. The very first one is always methionine, methionine. Methionine is the amino acid that AUG, the start codon, codes for. So we're going to move over, we're going to read the codon. Once we've determined which amino acid is needed, a tRNA molecule is going to bind that amino acid and deliver it to the ribosome. Now your tRNA molecules are the shortest RNA molecules. Each one is an anti-codon sequence. And when I say it's an anti-codon, I mean that it's the complementary codon to the one that we just read. Following our standard rules of complementarity, wherever there's a G, we have a C, and vice versa. Wherever there is an A, we have a U, and vice versa. Remember, this is RNA, there's no T, we use uracil in place of thymine. So in other words, if my sequence is GAC in the mRNA, its anticodon would be a tRNA molecule that says CUG. So GAC, CUG. Remember, A pairs with U, G pairs with C. Now that anticodon has a specific shape that allows it to bind to a particular amino acid, whatever amino acid that the codon we read represents. The tRNA delivers that amino acid to the ribosome and then binds it to the existing chain. The activating enzyme is going to form a peptide bond between the two, linking up whatever amino acid we just brought to the preceding amino acid. We have now joined those two amino acids together to form a dipeptide, D-I-P-E-P-T-I-D-E. Di means two, dipeptide, two amino acids joined together by a peptide bond. We've begun assembling our polypeptide. Once this has happened, what's going to happen is that the ribosome is going to translocate. What translocation means is that the tRNA molecule is going to detach from the amino acid, go away, it doesn't need to be there anymore, and then the ribosome is going to move down and read the next codon in line. And we are going to repeat this cycle. The ribosome moves in the 5' to 3' direction, it reads a codon. We attract a tRNA molecule with the anticodon sequence. That tRNA brings along the amino acid that the original codon codes for. We attach it to the existing polypeptide chain, form a new peptide bond, and then we translocate again. tRNA gets out of the way, ribosome moves on to the next codon in line. And we are going to repeat this process, slowly working our way down the mRNA molecule, reading one codon at a time, adding on one amino acid at a time. And little by little, we're going to find our polypeptide sort of dangling off the ribosome, kind of like a string of tickets dangling out of the skee ball machine when you keep playing one game after another and you don't pull the tickets off. All right, so we're going to keep on doing this, slowly working our way down. Remember, RNA is single-stranded, so we don't have a second strand to read. It's all going in the same direction. That's elongation. Now, sooner or later, we are going to hit the end of the mRNA. And that brings us to the final step, which is termination. When termination happens, termination happens when the ribosome gets to a stop codon. Remember that there are 3 possible stop codons, UAG, UAA, and UGA. The moment the ribosome reads any of those 3 codons, it's going to say, all right, we are done. We are no longer building new polypeptide. We're finished. Instead of attracting a tRNA molecule, when we read the stop codon, this is going to cause an enzyme called a release factor to remove the polypeptide we have been building from the ribosome. Now it is a separate unit. The ribosome then disengages from the mRNA, the complex that we built back in initiation, breaks down. Now what we have got is a single polypeptide molecule. The exact sequence, if all went well, that mRNA molecule was coding for. We've just expressed the gene. We have built the protein product that gene codes for. Now that polypeptide might by itself be a functional protein that can then go do whatever the cell needs it to do, or that polypeptide might just be a part of a larger protein. We might need to express a couple of different genes to build the polypeptides they code for so that all of them can come together and form the quaternary structure of that final finished protein. All right, now that is termination, and that is translation. We've just been through the whole process of translation here. Now, important thing to know about this. Most of the time, when your cell is expressing a gene, it doesn't need just one copy of that protein molecule. You're going to need lots of copies of that protein molecule to do whatever things you need to do. One molecule is not much. You might need thousands or millions of copies of that molecule. So what do? Well, a couple things can happen here. One is that when the ribosome is done reading that mRNA, whenever it finishes translation, it detaches from the mRNA molecule, and very often what will happen is it will go right back to the beginning, bind on to the start codon, and translate it again. The entire ribosome will move down the length of the mRNA, repeat translation, and build another copy of that same polypeptide. To further speed things up, it is not unusual for multiple ribosomes to be working on the same mRNA molecule at the same time. That you've got, okay, you build on, and then this ribosome moves down to read the next codon, and another ribosome attaches to the start codon behind it. And then they both move down to continue, and another one attaches to the start codon behind them. So it's not unusual to find a whole line of ribosomes, like train cars, working their way one after another along the same mRNA molecule at the same time, each one cranking out another copy of that polypeptide, and each one ready to go back to the beginning and start over again once it finishes. In this way, we can greatly accelerate the rate at which that polypeptide is produced to help the cell meet its needs sooner. In some cases, the DNA will express a given gene more. There are rates of genetic expression. Some genes are expressed more frequently at certain points in the life cycle than others, and that might mean that back inside the nucleus, we are once again transcribing that same gene. We're making another mRNA copy of that same gene that can go out and then be translated by multiple ribosomes at once to further speed up the rate at which that protein is produced. So, by making multiple mRNA transcripts of the gene, by having multiple ribosomes working on each one simultaneously, and by having each ribosome go back and translate again when it's done, we can greatly speed up the rate at which we produce the needed polypeptide. And in that way, the cell can meet its needs. A lot of times this is regulated by feedback inhibition, which you might recall way back from our discussion about enzymes, that the process is regulated by the concentration of the needed polypeptide. And whenever it hits a certain concentration within the cell says, okay, we're done. We have enough of this that we need right now, and the process shuts off. Now, once that is finished, once we're done translating, once all of this is over, what's going to happen is that the cell is going to digest the mRNA molecules. We're going to break down those mRNA transcripts of that gene using enzymes called RNases, capital RNA, lowercase ase. RNases. Those are the enzymes that digest RNA. We're going to break down those mRNA molecules once we don't need them anymore, and then the nucleotides from them can be recycled. We can take those nucleotides and use them to build other RNAs that the cell needs. Not only other mRNA transcripts, but also rRNA for ribosomes and tRNA for the process of translation, which have their own methods of being built that we're not going to go into right now. So that, from beginning to end, folks, is gene expression. We have just transcribed a gene from DNA to a molecule of mRNA. We've copied out the gene that we want to express. That mRNA molecule has traveled to a ribosome and it has been translated, meaning that the ribosome has read it and built the polypeptide product that it contains the code for. Now that protein or polypeptide is available for the cell to use and carry out whatever function that gene is the gene for. So if I say, here is the gene for lactase, I've expressed it and I've built the lactase enzyme, which can now go and do the job of digesting milk sugar. All right, it's all sounding good. Okay, now let's talk about a problem. When DNA was first identified, when we started learning about codons and started learning about genes, in the earliest days of genetic science, we thought one gene equals one protein. That was the general belief about how this works. We later learned that it was one gene equals one polypeptide, that sometimes you needed multiple genes to build the same protein. Now the assumption was that there were going to be approximately 155,000 genes present in our DNA. Reason being because we know of about that many polypeptide products that our bodies produce and about that many different mRNA molecules that our cells produce. So we figured, okay, one gene, one polypeptide. Each gene codes for one particular thing. There are 155,000 polypeptides in the human proteome. Ergo, human DNA must contain about 155,000 genes. That was the general belief in biology for a long time. Well, the Human Genome Project began in 1992, the efforts to sequence the entirety of the human genome, by which I mean looking at human DNA, identifying how many genes there are, and where each gene goes in the DNA. What is the locus of that gene? If you don't know that word, L-O-C-U-S, the locus is the specific location in the DNA where a given gene is found. That is always the same for all of us. The gene for eye color in me is found on the same chromosome in the same spot that it is in you. That's the locus for that gene. So the goal of the Human Genome Project was to map the entirety of the human genome, all of the different genes that are possessed by human beings, basically figuring out how many genes people have and where in the body they are located. Actually, Human Genome Project began in its early organizational stages in 1988. James Watson began working on it in 1992, same guy who worked on other aspects of the central dogma and the structure of DNA in the past. Another team of scientists led by J. Craig Venter, J. That's, he just goes by the initial J rather than his first name, Craig Venter, V-E-N-T-E-R. J. Craig Venter was leading a separate team of scientists on the other side of the country who were also trying to sequence the human genome. It was estimated that the whole project was going to take like 20 years to do with the technology we had in the 1990s. Well, in the early 2000s, Venter and Watson got together and said, you know what, we should not be doing this separately. Let's team up, let's combine our expertise and our technology and see if we can sequence the human genome faster so that medicine and genetics and other fields that need that data have access to it sooner. Together, Watson and Venter were able to complete the sequencing of the human genome in just 5 years. And so as of the early 2000s, we now had finished sequencing the human genome. We had a complete map of human DNA. Now, that did not automatically mean that we knew what every gene was for. It meant we knew where every gene was located in the DNA. It did not automatically tell us what each and every gene did. We're still learning that. And since then, we've sequenced the genomes of lots and lots and lots of organisms. It's not hard to sequence genomes nowadays. The technology has improved to the point where what used to take years to sequence a whole genome can now be done in days. So sequencing the genome of a species is no longer that hard, and learning all of that has really helped us in a lot of areas. It's helped us to identify what genes are responsible for specific genetic disorders. It's helped us to compare the genomes of different species to learn things about how organisms are related to each other and how and when they diverged from each other in the pathway of evolution. And so it's been an immensely, immensely helpful thing for both medicine and for biology as a whole. So here's the thing that was the surprise. When we finished sequencing the human genome, the big surprise was that we found out there were way fewer genes than anyone thought. We had suspected about 155,000 genes in the human genome. It turned out it was closer to 25,000. Way less than we thought, like 1/6 as many genes as we expected to be there. And the field of genetics was floored by this. It's like, okay, how are we coding for so many different polypeptides when there are so few genes to make them? How can our bodies make 155,000 different protein products with only 25,000 genes to do that? Well, the answer to that lies in this thing I skipped over before, that I told you that between transcription and translation, there was another thing that needed to happen, a step called splicing. And splicing is going to help us to answer that question. But bear with me while I tell you the story. Now, if you don't already know this, much of your DNA is not coding for anything at all. You've already heard about several non-coding regions of the DNA. You've heard about the origin of replication that binds the enzymes needed for DNA replication but doesn't actually contain the code for any genetics. You've heard about the promoter, which does a similar job for RNA transcription and is found at the beginning of every gene. You've heard about the termination site and the terminator that are both non-coding areas that tell our cells when to stop DNA replication and when to stop RNA transcription. And there are a bunch of regulatory areas of the DNA that I haven't talked about in here. Take a genetics class and you'll learn about them, that have other miscellaneous functions in not necessarily coding for genes, but in telling the DNA when to express them and how. Well, what I have also not told you is that a significant percentage of the DNA is not coding for anything, or not as far as we can tell. A lot of our DNA is not obviously a part of any gene. Those non-coding regions of the DNA are called introns, I-N-T-R-O-N-S, introns. Coding regions of the DNA are called exons, E-X-O-N-S. Those are the regions that actually contain the functional genes that we've been talking about. Now, where do introns come from? Some of them are doing other jobs other than coding for stuff like, origins of replication. Some introns are leftovers from ancient mutations. They're situations in which genes that were part of you at one time ceased to be functional. They're vestigial genes remnant from earlier stages of our evolution that are still present in our DNA. They're just not being expressed, or they are defunct genes that got mutated to the point that they couldn't do anything anymore, and they're just kind of sitting there now. Because there's no automatic mechanism in your body for removing a gene from DNA when it's not doing anything. In fact, all the mechanisms we learned about in DNA replication that are there specifically to make sure that DNA is faithfully copied also have the effect of conserving defunct genes as well, and therefore they don't get removed from the DNA automatically. That's actually really useful because we can look at those mutated sequences, compare them in different species, and we can do molecular clocking to calculate how long ago those species diverged, I should say, from a common ancestor, and therefore track out the pace of evolutionary time. Also reveal things to us about how one organism is related to another one in ways that might not be obvious because they don't look much like each other anymore, but they have common genetic sequences that show that connection. Some introns are the result of viruses. Certain viruses function by attaching themselves to your DNA and just kind of going along for the ride. We're going to talk more about this when I do a lesson on viruses at the end of the semester, and not all viruses work that way. The cold, the flu, the coronavirus, those don't work that way. But certain viruses, like HIV, actually attach themselves directly to your DNA and get copied along with the rest of it. They're fiendishly hard viruses to fight because there's no real way to kill them without destroying your DNA as well. At least not that we found thus far. Now here's the thing, you think virus, you think getting you sick. There are lots of viruses that don't make you sick at all. There are lots of viruses that the only thing the virus does is get copied along with the rest of the DNA. And once they bond onto your DNA, they're just basically sitting there, getting copied. Anytime the DNA gets copied, the virus is not doing anything, bad or good. It's just there. It's just part of your genetics now. It's a set of introns, viral genes that are basically non-functional. We actually have more viral genes in our DNA than we have human genes in our DNA. By some estimates, we have 30 to 35,000 viral genes, most of which are not active at all. So there are a lot of possible sources of introns. And there are some sections of the DNA that are just, we don't know what they're there for. They're just bits that are clearly not coding for anything, but they're just strings of letters that are present. Are they the result of old errors? Are they a result of defunct mutations? Are they doing something else? A lot of it is a question mark. Biologists used to call it junk DNA. Well, we have since learned the story is a little more complicated than just saying, oh, this is non-functional DNA. Here's why. Whenever you are copying a gene during transcription, you start at the beginning of the gene, you work your way to the end of it. The RNA polymerase starts at the start codon of that gene, goes all the way down to the end of that gene, and it copies everything between start codon and stop codon. The entire sequence of that gene beginning to end. That's what we want, right? We want to get all the instructions. Here's the thing. I told you that we have introns, which are non-coding regions, and exons, which are coding regions. What I did not tell you is that they're not nicely grouped together. It's not like, okay, here are all the exons here, and here are all the introns over there. The introns are scattered throughout the exons. They are distributed among them. In other words, from here to here contains the whole sequence of the gene I want to copy, but in the middle of it, you're going to find chunks of introns that it's broken up by non-coding regions. That's not just, okay, a nice unbroken gene from beginning to end. The gene is in fact broken up. It's broken into several sections with introns dividing up the exons from each other. In fact, there are actually more introns than exons. Your genome is about 98 to 99% introns. Only about 1 to 2% of your DNA is actually coding regions or exons. So there's more intron than exon between point A at the beginning of the gene and point B at the end of the gene. But you know what? During transcription, we just copied the whole damn thing. We started at the beginning of that gene at the promoter, we worked our way to the end at the terminator, and we copied everything between those two, introns and exons. So when we are done with transcription, the molecule that we have does contain all the instructions that we need for the protein that we want to create during translation, but we also have a lot of junk in there. We also have a lot of non-coding regions interspersed among the exons, which are not part of the gene that we want to copy. The transcript, in other words, needs editing. This is why sometimes the molecule that we make at the end of transcription is referred to as pre-RNA, because it's not quite ready for translation yet. Before we can translate that gene and build the protein that it codes for, the ribosome is dumb. The ribosome cannot look at the gene and say, that there is an intron, I can just skip over it, let's move on to the next exon. The ribosome just moves down the gene from one end to the other, and every time it sees 3 nucleotides, it reads them and builds another amino acid on. We can't have it doing that with the introns, or we're going to have a genetic sequence that makes no sense. We're going to have random amino acids shoved in there that are not part of our polypeptide. So, before we can translate this pre-RNA molecule, we have to remove the introns. We have to take out the non-coding regions that were copied along with the coding regions. That is going to involve a process called splicing. S-P-L-I-C-I-N-G. When you splice a gene, you are going to cut it to pieces and then stitch it back together in the way that you want it to be. We use the word splicing a lot in genetic laboratories. It's a thing we can do artificially whenever we're studying DNA and RNA in lab environments. We can use RNase, that is RNA digesting enzymes, and DNase, or DNA digesting enzymes, to break apart DNA and RNA into smaller fragments for laboratory study for a wide variety of purposes. Some of you may have looked at some of those in your lab sections of the course. Things like, DNA fingerprinting, for example. But we're not going to get into that functional stuff right now. Our interest is that right now we have an mRNA or pre-mRNA molecule that has exons for the gene we want to express, but it also has a lot of crappy introns in there that we don't want to be part of it. So the process of splicing is going to be the process by which the cell cuts apart that pre-mRNA molecule, removes the introns, and then puts the exons back together to make a complete mRNA that is ready to be translated. Here's how it works. Splicing happens in the nucleus, at least assuming that you're a eukaryote, it happens in the nucleus. And what's going to happen is that there are regulatory molecules in the nucleus who govern this process. Those molecules are called snRNPs. That's lowercase sn, capital RNP. By the way, that stands for small nucleic ribonucleoproteins. They're a combination of RNA and protein. snRNPs, lowercase sn, capital RNP, which we often call in biology by the absolutely wonderful name SNRPs. So you'll hear them called SNRPs. Sounds like characters from like an old 80s cartoon show. But anyhow, though, the SNRPs are the regulators of splicing. We do not fully understand the mechanism yet. This is a thing that has not yet been fully solved in the biosciences, but somehow or another, the SNRPs are able to look at an mRNA molecule and say, that is an exon, that is an intron. The SNRPs can somehow distinguish introns from exons. Once the SNRPs have determined what is part of the gene we actually want to express and what isn't, they are going to direct an enzyme complex known as spliceosomes. Gotta love these names. S-P-L-I-C-O-S-O-M-E-S. Spliceosomes. The SNRPs are going to direct enzyme complexes called spliceosomes, which will go in, cut out the introns. They will remove the introns from the mRNA sequence. Once the introns have been removed, all the exon bits that are left, which remember might be only 1% of the whole transcript, are going to be stitched together. They're going to be spliced into one single unit. Therefore, at the end of splicing, what we now have is a much shorter mRNA molecule. And we can truly call it mRNA now rather than pre-mRNA. because all that's left in it at this point is the coding regions. All that is left is exons. And now it's ready to be translated. So that's what splicing is. Splicing is going in, removing the introns, and stitching the exons back together into a unit that only contains coding sequences. That is the thing that then goes to the ribosome for translation, because that way the only things we're translating are the actual instructions for the polypeptide that we want to build. And like I said, there are still things we don't know about the process of splicing. The way SNRPs work is not yet well understood, but this is what's happening. Now, let's loop back to the thing I said a while ago. I told you at the beginning of this little subsection of our discussion that we had discovered when we finished sequencing the human genome that there were only 25,000 or so genes responsible for the 155,000 or so polypeptides made by the human body. And so everybody's like, what the hell is going on with that? How can we make this many different proteins if we have so few genes to make them? Here's the thing we have learned. All right, so I copied my mRNA, right? And let me just draw a little quick diagram to show you how this might look. Okay, so let's imagine that that first line that I drew on the right, that is our complete pre-mRNA transcript. Don't worry about the other things I have on there, those are for other classes. But that's our complete pre-mRNA transcript from beginning to end. We copied everything, whether it was an intron or an exon. The areas that I have filled in on that line are the exons. Those are the coding regions. And you can see most of what we copied was introns. During splicing, we're going to cut out the introns and we're going to stitch back together the exons into a much smaller final mRNA molecule consisting exclusively of coding regions. That's how we splice that. Now we can go translate that gene, right? How about this? Let's do something a little different. Let's take that exact same mRNA sequence, only we're going to splice it in a different way. Let's take that exact same mRNA from beginning to end, but wherever I X'd stuff out, let's leave that stuff out of the transcript. Let's splice it a different way. Take that same mRNA sequence, and we're going to leave out a couple of bits that I put X's on that normally would have stayed in as exons, but we're going to take a couple of areas of introns, the ones that I circled, that were not part of the original, and we're going to leave those introns in. In other words, we're going to splice that sequence in a completely different way, and the final thing that we get at the end will be a different mRNA pulled from the same gene. In other words, what we have learned is that you can splice the same genetic sequence in more than one way. We got gene that begins here, gene that begins there. Okay, that's whole sequence, cut out the introns, make a much smaller little mRNA that's all exons. But take that same sequence and splice it differently. Leave in that bit there that we otherwise would have cut out. Leave out that bit that we left in last time. And let's leave in two more little bits over here. And now we're going to splice it in a slightly different way. Same gene beginning to end, but by splicing it differently, we get a different mRNA at the end. In other words, we can get more than one possible polypeptide from the same gene, depending on how we splice the pre-mRNA. Any given pre-mRNA molecule can be spliced in different ways to give us different mRNAs at the end, which when translated would yield different polypeptides. To put it another way, one of our assumptions about genetics seems to be wrong. I told you guys back when we talked about DNA that it was one gene equals one polypeptide. that every gene is the instructions on how to assemble one single polypeptide. We have now learned it's not that simple. That any given gene in your DNA might only contain the instructions for one polypeptide, but it's possible that that gene might be able to be spliced in a different way to produce a completely different mRNA, and therefore to produce a second polypeptide from the same genetic sequence, or a third, or a fifth. The truth is, right now, we do not know what the maximum number of different polypeptides is that you can get out of the same gene by splicing that gene in different ways. But given the fact that we have about 1/6 as many genes in our DNA as there are known protein products produced by your body, it is abundantly clear that most of our genes are producing multiple different polypeptides. They're coding for several different things, all depending on how you splice the pre-mRNA. In other words, folks, genetics is a lot more complicated than we initially thought. We cannot say one gene equals one polypeptide. It is obvious that one gene might actually equal several different polypeptides, depending on what bits you leave in and what bits you leave out. In other words, what is an intron or an exon may depend on what protein we're trying to build. And when I have that sequence from beginning to end, maybe this bit right here is an intron when I'm coding for one polypeptide, and we'll cut it out and it won't be part of it. But that bit right here is an exon when we're coding for a different polypeptide. And you leave it in, and you'll get a different protein product after translation. This has been one of the more exciting discoveries in modern genetics. It's also been one of the more frustrating discoveries in modern genetics, because we can no longer look at a gene and say, okay, we have determined that that gene is the gene for X. Well, we can say that, but that doesn't tell us whether that gene might also be the gene for Y and Z and A if we splice it a different way. So the task of assigning what each gene does has become a lot more complicated, because any given gene might realistically have more than one thing that it codes for. And we don't have an upper limit on how many things it might code for. We don't know what the maximum number of things that a given gene can code for is. And so the science of mapping out our genome completely and determining what each gene does has gotten a lot more sophisticated because of this discovery. Okay, folks, but that, I think, gives us everything that I want to tell you right now about gene expression. We have now learned for our chapter on DNA how DNA stores data. And between these two lessons, we have now learned how we copy that data from the DNA to the RNA during transcription, how we splice that data to remove non-coding regions, and then how we translate the final mRNA molecule to make the protein that your cell requires. All right, that's gene expression, that's RNA. In our next section, to finish out our look at DNA and RNA, we're going to talk about mutation. We're going to talk about what happens, because everything I've been discussing thus far tells you what happens when this goes right, when everything happens the way it should. In our next lesson or two, we're going to look at what happens when things go wrong. What happens when the DNA gets damaged. That's a thing called mutation, and we're going to learn about what the consequences of mutation might be, including a consequence known as cancer. All right, so I think that's a good point to leave off. I hope you all have a good afternoon. Thank you.